Hi, welcome back to Protein Function in Biochemistry. My name is Kevin Tokoph. Make sure to like this video and subscribe to the channel for future videos and notifications. In this video, we're going to continue our discussion of hemoglobin, and we're going to actually more specifically continue our discussion of the Bohr effect, which we went into in the last video. If you need a review of the Bohr effect, certainly go back and watch that. I'll briefly talk about what it is. And the concept of the Bohr effect is basically summarized right here by this molecular equation, and then also this right here. And what it says is that whenever you have an area in the body, the blood that is, because that's where hemoglobin is, when you have an area of low pH, that corresponds to a high hydrogen ion concentration, and that also corresponds and tends to correspond to a high carbon dioxide concentration. When these three dependent conditions are met, there's a tendency for the release of oxygen from hemoglobin to the tissues. Okay. The opposite is also true. If I have high pH and low hydrogen ion concentration and low CO2, that tends to favor binding of oxygen. Okay. Now keep in mind that if I want release of oxygen to the tissues, I do want a low pH, which generally is going to correspond to a high hydrogen ion concentration, right? So that concept is explained by this equation. So remember I talked about that hemoglobin is written as HB, that's the abbreviation. And whenever I have anything bound to it, could be anything. This X could be anything. If it was carbon dioxide, if it was carbon monoxide, nitric oxide, or oxygen, I simply draw hemoglobin and put a dash and then whatever it is that's bound to it. So this is hemoglobin bound to oxygen, some amount of it. And this is going to be called oxyhemoglobin. Okay, so the idea is if I have high hydrogen ion concentration, okay, I'm simply going to ultimately get Le Chatelier's principle. This video is going to be largely about Le Chatelier's principle from general chemistry. So if I add a lot of hydrogen ions, I'm adding a reactant for this equation, and I'm essentially going to shift this to the right. Right? If I add one reactant in a large excess, I shift the reaction to the right. That's Le Chatelier's principle. What's on the right side? Well, I have the hydrogen ion bound to hemoglobin, but notice the oxygen is released. Okay, so in other words, whenever I have really low pH, that causes a shift, and this is the Bohr shift towards the right here, which means oxygen dissociation. Okay, and that occurs near the tissues. And we go over the rationale behind that and why that's important in the previous video. Here what I want to talk about is more the molecular mechanism by which this happens. Before we do any of that, we need to go over this chemical equation shown right here. This set of basically two equations, this is the buffer system in the blood. Okay, keep in mind hemoglobin is in the blood, and just like anywhere else in the body, it has to be buffered. This is called the bicarbonate carbonic acid buffering system. The bicarbonate, which is this right here, and here's the carbonic acid, that's where it gets its name, the bicarbonate carbonic acid buffering system. And it maintains the pH of the blood usually between about 7.35 and 7.45, and normally we say the normal pH is about 7.4. So whenever we talk about blood pH, I'm usually going to talk about 7.4 in humans. Okay? What does the equation say? It says that when I have hydrogen ions, they can combine with bicarbonate. That's what this is, bicarbonate anion. And they can form something called carbonic acid. So this is called carbonic acid. There's an enzyme referred to as carbonic anhydrase. If you actually want to see how this enzyme works in more detail, there's a video on that. Actually, just go to the YouTube search, carbonic anhydrase, and it's one of the top videos. And this enzyme breaks carbonic acid down into carbon dioxide and water. And we also talked about, recall, that anytime I have high hydrogen ions, it corresponds to high CO2. This is why. And it really operates through Le Chatelier's principle. For example, if I have a lot of hydrogen ions, right, by Le Chatelier's principle, I have a large excess of reactants in this equation, that's going to shift everything here to the right. And even though this is a set of technically two equations, the first one is to carbonic acid and the second one is to CO2 and water, it still functions in exactly the same way. If I load it up with this reactant, H+, it's going to shift it all the way to where I ultimately get a large amount of carbon dioxide. So this is ultimately, this buffer system equation, this is why when you have a low pH or high hydrogen ion concentration, it corresponds to usually in that area or vicinity a high CO2 concentration. Okay? So the first thing we're going to do is we're going to talk about explicitly why hydrogen ions have a tendency to cause oxygen dissociation from hemoglobin to feed the tissues. And then we're going to do a brief discussion of carbon dioxide, and that's also going to facilitate our discussion in the next few videos about the Haldane effect. And recall that the Bohr effect specifically, when we talk about the Bohr effect literally has to do with pH. Okay? It says, how does pH affect how oxygen binds to hemoglobin? The Haldane effect is almost the same thing, except it's more concerned with how does carbon dioxide affect how oxygen binds? And then conversely, how does oxygen binding affect carbon dioxide binding to hemoglobin? So Bohr effect is all about pH. How does this work? Well, in hemoglobin, recall hemoglobin is a tetramer. It has 2 alpha subunits and 2 beta subunits. Now, they're not these sizes, and these are certainly not proportional either. They're actually a lot larger. I'm showing some critical parts here. Of hemoglobin, this is one of the alpha subunits, and this is one of the beta subunits. All right. What I want to show you is that there's a certain amino acid residues in this protein that ultimately play a big role in whether or not oxygen is binding, if that's the tendency, or if the tendency is for it to be released. This condition right here, this is when we tend to have higher relative pH. In other words, this is when we have a lower concentration of hydrogen ions. So just bear that in mind for this set of pictures. All right. So what happens? All right. Right here, you can hopefully see this is a lysine residue. In fact, this one actually is the N-terminal lysine residue. We notice that it has an R group right here, and its R group is a protonated amine. It's going to be really important. Over here, this is actually, this is one of the amino acids. This is actually a histidine on the beta subunit. Okay, this is a histidine. And one thing that's really important is histidine has a hydrogen that's bound to a nitrogen here. And because hydrogen is less electronegative than this nitrogen, there's a dipole, and that causes this hydrogen atom to develop a partial positive charge. Also, whenever there is a higher pH, and notice that means generally a pH that's greater than 7.4, but we're also going to say pH less than 7.45 because remember, it's buffered so it doesn't get above that. It's basically, when we look up here, it's on the upper end of this range. So a higher relative pH. It causes this carboxyl group of this histidine, because this is actually, this is the C terminus over here, okay? It causes it to be deprotonated. Okay, remember if I have a general carboxyl, it could either exist in the protonated state or the deprotonated. If it's protonated like this, then there's no net negative charge here, so that's not what happens. Okay, but when there's a higher relative pH, it turns out that this carboxyl is actually in the deprotonated state. Okay, also over here, this is actually an aspartate residue, and this R group over here is its carboxyl from the R group, and it's also in the deprotonated state. Now notice at higher pH, when there's lower H+, this imidazole of the histidine, this partial positive, can form an electrostatic interaction with the R group of this aspartate, that is this carboxyl. Also, the lysine over here on the alpha subunit can form an electrostatic interaction with the negative charge of the C-terminal carboxyl of the histidine on the beta subunit. Okay? And so the basis for the binding of oxygen are these primary electrostatic interactions. Okay? And it doesn't seem like much, but when there is higher pH, these electrostatic interactions do form, and that tends to favor binding of oxygen. Now, one thing that is really important here is that binding of oxygen occurs in one of the 2 states of hemoglobin. Now, there are 2 states. There is an R state and a T state. Okay? The R state is basically whenever you are releasing hemoglobin, okay? The R actually stands for relaxed, and basically that is when oxygen tends to be bound. The T state stands for tense, and the T state is when oxygen is dissociating more than binding. So if you had to compare R versus T state for this is binding of oxygen, so this is actually more in the relaxed state, okay? So ultimately, when you have these electrostatic interactions at a higher pH or low H plus concentration, it tends to stabilize hemoglobin in the relaxed state or the R state, okay? And this is when binding of oxygen is more favored, okay? That's very important. There's another thing that also happens because when you have these electrostatic interactions, remember, this is not exactly where the oxygen is binding. Oxygen binds at heme, but these electrostatic interactions tend to make the area around the heme itself more relaxed, and that's ultimately what makes the oxygen bind better. Okay? Now, let's do a case where we shift it this way to lower pH. So now, over here, I'm raising the concentration of hydrogen ions. Okay? So it turns out that these amines right here, so this amine here on the lysine, and the imidazole nitrogens here, they're not really affected by lowering the pH. Notice I still have the protonated amine here of the R group of this lysine. The imidazole here did not change at all. There is still a partial positive on this hydrogen, but here's the problem. Whenever I do lower the pH, and I raise the H plus concentration, these carboxyls, the one here on the histidine, this carboxyl, and the one here on the aspartate R group, they get protonated. Now the question I go back to is, okay, which one of these carboxyls actually has a negative charge? Well, this one is protonated, so it's neutral. This is the only one with a negative charge. So if I protonate this, there's no more negative charge. So is there going to be a sufficient interaction between this carboxyl and the lysine R group? The answer is no. There is no interaction here. I'll say no significant interaction. Now let's look at this. This aspartate R group, this carboxyl, also gets protonated. Is there here any interaction between this partial positive and this? The answer is, again, no. There is no significant interaction. So in the case of higher pH or lower H+, you have these, and that tends to stabilize it in the R state. But here, when you lose those electrostatic interactions, you actually tend to stabilize this in the T state. And it's specifically the T state with respect to where the oxygen binds, or where it would normally bind. And the T state is tense, and the T state tends to favor the release of oxygen. Okay, we're going to go into a little more detail on T versus R state in another video. But suffice it to say, the T state you tend to get release of oxygen, and for the R state you tend to get binding of oxygen. Now here's sort of an obvious question to think about. Where in the body might it be important to stabilize something in the R state, where you have binding of oxygen, versus where might it be more important to stabilize it in the T state, where you have release of oxygen? Well, where do you want release of most of the oxygen? By metabolically active tissues, right? That's where you want it, okay? So, if you want it near metabolically active tissues, remember, metabolically active tissues produce a lot of hydrogen ions, where you get lowered pH. What do you have here? You have lowered pH. These carboxyls get protonated, it stabilizes it in the T state, and you get release of oxygen, because metabolically active tissues, they need oxygen. Very important concept. And hopefully that makes sense. Now, on the flip side, where do you want binding of oxygen? Well, you want binding of oxygen generally to occur wherever you're picking up oxygen. So think back to your general biology, or A&P, or physiology. Where in the body does hemoglobin pick up oxygen? It picks up oxygen in the lungs, right? So perhaps maybe you would want the binding of oxygen to occur in the lungs. So maybe in the lungs you'd want this to be more in the R state, okay? So that's something we're going to be thinking about as well. Okay, so you're going to want this in the R state, okay, in the lungs. And in another video, we're actually going to go over another mechanism by which that ultimately occurs. Okay, it turns out there is a modulator between the R and T states, and it's called 2,3-bisphosphoglycerate, or 2,3-BPG. It's an allosteric regulator of hemoglobin, and we're going to discuss that more in a separate video. Okay? All right, here's another super important concept that we're going to do before we conclude this video. Remember I mentioned a little bit ago, the very beginning of the video, that through this Le Chatelier's principle of the buffering system of the blood, if I have high hydrogen ion concentration, that's going to correspond ultimately to high carbon dioxide levels. Well, it turns out that carbon dioxide actually also tends to favor a release of oxygen. If you remember, and I talked about this in the previous video, that whenever I have high hydrogen ions near tissues, there's also a tendency for there to be high carbon dioxide. Carbon dioxide is a waste product of metabolism for cells. So the carbon dioxide, along with the H+, signals to the hemoglobin that I'm a metabolically active cell, tissue, I need oxygen, so it would make sense that the carbon dioxide would ultimately signal to release oxygen. Let's look at how that is. Okay, on hemoglobin, there are lysine residues. This is a lysine. Okay, this is one lysine. And you have a carbon dioxide molecule floating by. Okay, and I won't get too much into the mechanism of this ultimately, but suffice it to say, if you were curious, remember, lysine can be fully protonated, but it is in equilibrium with its deprotonated form, with the lone pair. So there is a chance this carbon dioxide will run into this nitrogen lone pair on the lysine, and when it does, it'll be nucleophilic attack on the carbon, and there will be a proton rearrangement, and what you ultimately get is this moiety right here attached to this nitrogen of the lysine on the hemoglobin. So this over here, this literally is just hemoglobin. This is a residue that comes off of hemoglobin, it's lysine, but when carbon dioxide reacts with it, this ultimately becomes a carbamino group. A carbamino group. Some textbooks will call it a carbamate, but technically this is a carbamino group. Some will call it a carbamine. And so effectively what happens is this carbon dioxide ultimately gets bound to the hemoglobin. So if you took A&P or physiology, one of the things you generally talk about is how oxygen and CO2 travel in the blood. You probably have heard most oxygen is bound to hemoglobin, right? You probably have also heard that a lot of CO2, some percentage of it, is also bound to hemoglobin. This is actually how it's bound. So it's not actually bound directly in the form of carbon dioxide, it's actually in the form of a carbamino group. And whenever you have carbon dioxide bound to the hemoglobin through this way, in this manner, the tendency is to favor O2 dissociation. In other words, it disfavors oxygen binding. It tends to make oxygen dissociate more easily, ultimately to feed the tissues. Okay, so if I come back over here to what we looked at in the last video, this Bohr Effect graph, I can also extend this to when I have this curve for low pH, this also means high carbon dioxide, and for high pH, this means low carbon dioxide. So you see high carbon dioxide, we'll do kind of what we did in the previous video, but if I take any general pressure of oxygen here and just draw a line up and see where it bisects the graphs, what I can see ultimately, if I get this done quickly, is I can see that at low pH, the red curve, there's a lower percent saturation with oxygen. So what that ultimately means is I have more O2 dissociation, right? That's what we talked about in the last video with respect to hydrogen ions. If I have high pH, low CO2, this is less O2 dissociation. How do I know that? Because the vertical axis is percent saturation of hemoglobin with oxygen. Remember, there's 4 binding sites for oxygen on each hemoglobin because there's 4 heme moieties, right? So are 4 of them bound, 3 oxygens bound, 2, 1, or none of them? Well, you can see that here for the high pH or low CO2, there's a much higher percent saturation with oxygen. There's more oxygen bound, which means less is dissociated. Here with low pH or high CO2, there's a lower percent saturation of oxygen, which means more of it has dissociated. So when we talk about carbon dioxide in the sense of the Bohr effect, because H plus corresponds to a high CO2 concentration in a particular vicinity of the blood, it turns out that low pH and high CO2 mean more oxygen dissociation, and high pH and low CO2 means less oxygen dissociation. And ultimately what it's all about is whether or not you stabilize this in the R state, which is what is shown here on the left. This is where binding of oxygen is favored, or in the T state where release of oxygen is favored. Okay? So hopefully this gives you a pretty good intuitive sense of the Bohr effect and the molecular mechanism behind why it works. In the next video, we're going to talk about the Haldane effect and do a very quick comparison of the Bohr effect versus the Haldane effect. And hopefully you'll get a complete picture of how hemoglobin works. After that, we'll go over an allosteric regulator of hemoglobin, which is called 2,3-bisphosphoglycerate or 2,3-BPG. So thank you for watching. Make sure to like this video, subscribe to the channel for future videos and notifications. Thank you very much.